Hi guys, how's the connection today? Can you hear me well?
Yes.
Yes.
Thank you. You know what? When we talked on Monday, I forgot to record the lecture. And you guys did not remind me. Not that I was supposed to, but nobody noticed that we were not being recorded. So I don't know what to do now. Now I have to basically do the lecture by myself and upload it again. So, bummer. Anyway, Wednesday, November 11th, 11 o'clock. We are on chapter 10. We are on chapter 10. There's somebody's chat. Yeah, the other section's lecture. Well, you know, I have thought about it. The problem is there's some information for the other section, like there's some dates and stuff that I sometimes tell students when the homeworks are due and stuff like that. And I don't want to create any confusion so that the students tell me, oh, you told us that the deadline was this, but the deadline was from the other section. So what I'll do is I will go over, and also the other section, they have shorter lectures. So their lectures are 15-minute lectures. So I will have to go over, thank you. I will have to go over those lectures and see if I can use them. All right. So we are on chapter 10. Let me start sharing. Start sharing. And we are discussing glycerophospholipids. We finished by discussing waxes, right? Is that correct? Is my recollection similar to yours?
Yeah.
Yes, I think so.
Waxes. All right, so let's talk about glycerophospholipids. Glycerophospholipids and other components of other types of lipids which compose cell membranes. Cell membranes will be the whole chapter by itself, chapter 11, and we will do that starting on Monday. All right. So primary constituents of cell membranes are these glycerophospholipids. Two fatty acids. So now hold on a second. Yeah. Okay. So so we'll have so these glycerophospholipids, they will be formed from glycerol, which is the main basic motif in the glycerophospholipids, and the two hydroxy groups, positions 1 and 2, these ones, they will be esterified. There'll be esterified, we'll see that in a moment, with long fatty acids. And the third one will form a phosphate ester. So now this is monoester, remember, as opposed to the phosphodiester that was on the test. So this is monoester, and you can see there are different ways to draw this, but this is this has a stereocenter. So this particular glycerol 3-phosphate, so this one is L. This one is L. So it's a chiral molecule because the 4 substituents are all different. And obviously the phosphate group is charged at physiological pH, right? pH of 7. The phosphate is deprotonated. All right, so so this is the general structure of these glycerophospholipids. So in general, there is one saturated fatty acid, and one slightly longer unsaturated fatty acid, which is usually attached to oxygen 2, and the saturated fatty acid is attached to oxygen 1. And then the head group phosphate is usually attached to something else, which we refer to as head group substituent. So this whole thing will be the head group, including the phosphate. Head group. And this will be the head group substituent. All right, so what kind of head group substituents we can have? So we can have no substituent, just a hydrogen, in which case the glycerophospholipid will be phosphatidic acid. Phosphatidic acid. And then there are different kinds of substituents. So usually they're polar. So for example, you can have this ethanolamine. Now, why is it ethanolamine? Is because it comes from this structure. Plus, and there is OH. And remember, ethanol is the alcohol that has hydroxy group and 2 carbons. And now there's an amino group. So that's why it's called ethanolamine. So phosphatidyl ethanolamine, phosphatidylcholine. So you know choline. So this should have a positive charge. Phosphatidylcholine. Now phosphatidylserine. So serine is an amino acid. So this will be neutral, right? So it has positive charge and negative charge at physiological pH. So the net charge is the charge which is attributed to the, including the phosphate, right? So keep in mind there's a phosphate here too, right? And the phosphate has 1 negative charge already. 1 negative charge already. And so if there are these 2 charges cancel each other out, there's still 1 negative charge from the phosphate, so it's -1. So here it's minus and plus, net charge 0, right? All right, so now you can have phosphatidylglycerol. Phosphatidylglycerol, so this is not charged, but the phosphate is charged, so negative charge, -1. Now there is slightly more complex compounds. So for example, this one is myo-inositol bisphosphate. So can't you remember if I told you about inositols or I told it to the other section? So inositols are sometimes referred to as pseudosugars. So remember, in a sugar, you have an oxygen in the ring. In inositols, these are polyhydroxylated cyclohexanes. So they are like sugars, but they're not exactly because there is no oxygen in the ring, so-called endocyclic oxygen is missing. So it's a cyclohexane with 3 hydroxy groups. Now, one of these hydroxy groups will be attached to the phosphate and 2 others will have phosphate of their own, right? And so total, there will be 3 phosphate group associated with these molecules. Now, one of the main function of these molecules is in signal transduction, right? So these phosphatidylinositol 4,5 bisphosphates, they actually cleaved oftentimes from the cell membrane, travel inside the cell, and deliver the signal. For example, inositol triphosphate goes to endoplasmic reticulum and signals the release of calcium from the ER, endoplasmic reticulum. Now, cardiolipin is a much more complex lipid. So just to show you that there are possibilities. Now there is a phosphate on this side, and then there is also stuff on this side, right? Also with the R1 and R2. So, but it's much less important, this type of lipid. All right, so some of the most common type of lipids, one is phosphatidylcholine, so shown here. A major component of most eukaryotic cell membranes, also prokaryotes, including E. coli. Prokaryotes, including E. coli, cannot synthesize this lipid, and so the membranes actually do not contain phosphatidylcholine. Take it back. So this is a major component of eukaryotic cell membranes, so distinctive feature of that. Another types of lipids are ether lipids. So far we talked about ester lipids. You can see there's ester bonds, right? So the fatty acids are attached to the glycerol using the ester linkages. You can also have ether lipids. And so, for example, this one is, just to show you the structure, you can see this one is actually, so there's no carbonyl, right? So there's a carbon oxygen bond. It's an ether type, and specifically vinyl ether. Specifically vinyl ether type structure. And now, so this one, so it's a aliphatic ether analog of phosphatidylcholine, right? So this one does not have a double bond. This one does not have a double bond. And this one actually has an acetate ester position too. But this one is an ether. So remember ethers, right? Ethers as opposed to esters. So which ones do you think are hydrolytically more stable? Ether lipids or ester lipids? Which one will undergo hydrolysis more rapidly?
Ester lipids.
Nah, ester lipids are quite, oh, sorry. Did I say rapidly? You're right, sorry, I'm sorry. Correct. So ester lipids are susceptible to attack by water, right? And so ether lipids are more stable. Ether lipids are more stable. So the water will not attack this carbon because this will be a poor leaving group. All right, poor leaving group. All right, so so for example, this ether lipid shown here is known as a platelet activating factor, right? So what do platelets do? Do you know what kind of cells they are and what they do?
They're thrombocytes and they stick together for clotting purposes.
Yeah, so they're blood cells, right? And they're undergo aggregation. And when there is a damage to a blood vessel, there are various activating factors released by the endothelial cells of the blood vessels, right? And one of them is a platelet activating factor, which will be released from the cell membrane, travel towards the platelets, stimulate the aggregation, and the platelets will block the hole in the blood vessel. So it plays a role in mediation of inflammation. All right, so these are glycerophospholipids. So the second main type of membrane phospholipids are sphingolipids. So the backbone of sphingolipids is not glycerol. So that's the main difference. And the backbone is actually amino alcohol sphingosine. Sphingosine, which is shown here. So this orange color, right? So you can see it looks like it's almost like a glycerol type structure, but not quite, right? So we have actually 2 hydroxyls at positions 1 and 3, and amino group at position 2. And then there's a fatty hydrocarbon tail, right, with a double bond in it. So this is kind of allylic alcohol here. So the carbohydrate tail is not attached to the hydroxyl. It's actually attached to carbon 3, right? So hydroxyl is actually free in sphingolipids. All right, so this will be sphingosine. Now there is also possibility now to make an amide with this nitrogen, right? And so if once you make the amide with this nitrogen, now this becomes, so it has now 2 hydrocarbon tails, a shorter one and a longer one, right? And then there's a head group. Now, if the head group, if there's nothing, hydrogen, so the sphingolipid is known as ceramide. Ceramide. Now, if instead of hydrogen, again, you have colon, so the type of sphingolipid is known as sphingomyelin. Sphingomyelin, so you can have glucosyl, cerebroside, for example. In this case, the head group will be molecule of glucose. Now you can have a globoside. Globoside when you have ditri or tetrasaccharide, right? So basically small saccharide. And there's also much more complex oligosaccharides, specifically containing a type of sugar known as sialic acid. So these are known as gangliosides. And so there are different, there's a naming system for gangliosides depending on where sialic acid is found in this oligosaccharide residue and how many of sialic acids are actually present, right? So this is something you will need to remember, but it's pretty easy. So start with sphingosine and ceramide, and then just a couple others, sphingomyelin, globoside, and ganglioside. I would remember these ones. Globoside, ganglioside, sphingomyelin, ceramide, and sphingosine. So let's see what else we have here. Amide linkage rather than ester. Now, what can you say about stability towards hydrolysis? Compare this to glycerophospholipid. So we talked ether versus ester. Now you compare it to what? So we compare an ester to what functional group in sphingolipids?
The amide.
Amide. So which one is more stable? And so you have an ester. So this is OChem2, right? You have an ester and you have an amide. And both of these can undergo hydrolysis, right? Attack here or attack there. So which one is more susceptible to this kind of hydrolysis?
Ester would be.
So ester, that's right. So yeah, so amides are much less susceptible. So similar, they're less susceptible like ethers. So sphingolipids, because of the amide functionality here and no ester here, they should be more resistant to acid, to base catalyzed hydrolysis, or even acid catalyzed hydrolysis, or just hydrolysis at neutral pH over a long period of time. So polar head group, sphingosine biglycosidic or phosphodiester linkage. And one thing to keep in mind, with this sugar that you have in glycosphingolipid is found largely in the outer face of plasma membrane, right? So these sugars, ditrio tetrasaccharide or sialic acid and gangliosides, so these will be in the outer portion of the leaf of the bilayer, lipid bilayer. And so as I mentioned to you, many of these serve as recognition sites, right? So many different cell types differ in terms of what kind of carbohydrates they have on a cell surface. And so that's one way, for example, for the biological systems to recognize what kind of cells these are, is by the type of carbohydrates which are exposed on the cell surface. And for example, sialic acid is expressed to a larger degree, for example, on the surface of cancer cells, right? So there are some strategies now to target cancer cells specifically by recognizing sialic acid on their surface, right? And so you can deliver a toxic agent, toxic drug, specifically to a cancer cell because it has sialic acid on its surface. All right, so these are sphingolipids. Now, one of the most common one is sphingomyelin. Sphingomyelin, so this one has sphingosine and amide, which has a linked fatty acid. Remember, this will be called ceramide, and then you attach the phosphocolon. So attach the phosphocolon, then you get the lipid known as sphingomyelin. So sphingomyelin is abundant in myelin sheath. Myelin sheath that surrounds some nerve cells in animals, right? So you know that so nerve cells in central nervous system, CNS, and the peripheral nervous system, these neurons are not exposed all by themselves, just like electric wire, right? Electric wire is always insulated. And part of the reason why it's insulated is not only so that we don't touch it and get electrocuted, but also it propagates the electric signal better, right? And the same with the nerve cells. So there's a myelin sheath which contains a number of different components, but one of them is the sphingomyelin. Sphingomyelin, which is a lipid which is produced in central nervous system by the types of cells known as glial cells. And in peripheral nervous system, they're known as Schwann cells. And these glial cells, they are unfortunately, actually, if they become cancerous, they give rise to a horrible type of cancer known as glioblastoma. Glioblastoma, which is an incurable type of brain cancer. Highly metastatic within the brain. And for example, some of the projects in my lab, my research lab, are aimed at development of agents which can help combat glioblastoma in cancer patients. One reason why glioblastoma is so hard to treat, one reason is because it's actually hiding behind a blood-brain barrier. Remember this, BBB, blood-brain barrier. So brain is actually separated from the rest, from the blood vessels, for example, and the rest of the body by a very thick lipid layer. And so it's very hard to for the drugs to get through. And the second reason is that glioblastoma cells, they're resistant to apoptosis. Remember I told you apoptosis? Resistant. To apoptosis, which is programmed cell death, and many drugs, many cancer drugs actually induce apoptosis in cancer cells and make them die. But glioblastoma cells do not die. They actually have defects in their apoptotic machinery, which allows them to survive. All right, so that's about sphingomyelin. As you can see, very important lipid. Now, structure is similar to phosphatidylcholine. So we know that, right? Phosphatidylcholine. We talked about that. And this is sphingomyelin. So they have a similar head groups, just different type of the rest of the structure. Glycerol and sphingosine. Okay, so glycosphingolipids are also responsible for the differences in blood types among different individuals, right? So you know that there are actually different blood types. There is O, A, B, and AB. There are 4 blood types, right? And that's actually due to the differences in the sugar content of sphingolipids. Sphingolipids, particularly basically what type of sugar is located in the head group of glycosphingolipids. And so the sugars are actually, the chemistry of the addition of these sugars is controlled by types of enzymes known as glycosyltransferases. Okay, and so the enzymes are specific for a particular sugar, and so you can see here if you individuals that have a glycosyltransferase, for example, if there is no active glycosyltransferase, then you will have a blood type O, and specifically known as O antigen. So let's go here. So if this is glycosphingolipids blood groups. So sphingosine, fatty acid, right? So ceramide. Now then you have your sugars, right? And so here are their abbreviations. So you can see here in this oligosaccharide tail, you will have glucose, the yellow one galactose, blue square acetylglucosamine, yellow sphere, another galactose, and red triangle fucose. So, but there is nothing else here, okay? So there is nothing else. And so this is known as O antigen. So there is no glycosyltransferase that adds another sugar to galactose, okay? So it's O antigen. Remember, antigens are parts. Who remembers what antigens are?
What antibodies binds to?
Right. So so they are specific parts of foreign molecules, right? That are recognized by the host immune system, specifically the antibodies. And so if so people with no glycosyltransferase, no specific glycosyltransferase, will have the O antigen. O antigen, so basically nothing. Now, if you at this position, if you have a N-acetylgalactosamine, then the addition of this square turns this into an A antigen. Okay, this is O, this is A, and a B will be this galactose by itself. Galactose by itself, right? And so, so this gives rise to 4 different blood groups, right? So basically people with O, no antigen, A, B, or both, A and B. Both A and B can be also present when you have both glycosyltransferases. Okay. And so, so the way this works, you probably know, right? So if you have the group O, like I have group O, which is the blood type that cannot receive blood from anybody else, but can give blood to everybody else. So specifically, since there is no antigen, right, it's an O antigen, or basically no antigen, it has none of them. But because of that, it has antibodies to A and B. It has antibodies to A and B because it has no A and B antigens. That's why the antibodies can circulate in the bloodstream. Now let's look at let's look at A. So let's look at A. So A, blood type A will have the A antigen. Specifically, it will have this N-acetylgalactosamine attached to the sphingolipids on the surface of the red blood cells, And so it will not have the anti-A antibodies because those would destroy the A antigen, or will destroy the red blood cells with the A antigen, but they will have anti-B antibodies, right? So group B is the reverse of that. There'll be B antigen, specifically galactose at this position, right? Galactose at this position. So B antigen and anti-A antibody. So B cannot receive blood from A. Why? Because the B has anti-A antibodies and A has A antigen. So if you take the A antigen and add it to the recipient with the group B, then these antibodies will destroy that. So you will basically generate an immune response. And same thing here, right? So individuals with the blood group A cannot receive blood from the individuals with the blood group B because of the presence of the B antigen in the blood of those individuals, of the donors. Now AB, so here we have both antigens present, both N-acetyl galactosamine and galactose. And so because of that, there is no antibodies, but both types of antigens are present, right? So both types of antigens are present. And so A and B individuals can receive blood from anybody. It can, because they have no antibodies. But they cannot give blood to anybody because they have A and B antigens. They cannot give to A, this has anti-B, cannot give to B, this is anti-A, cannot give to O because it has anti-A, anti-B. This makes sense? So, all right, so these are ABO blood group antigens present on red blood cells and IgM antibodies present in a serum. Do you guys have any questions? Yeah, so you can see that something that was probably mysterious to you before this class, right? The blood types can be very easily explained using different carbohydrates whose structures you know, right? Whose structures you know, and how they form antigens and specifically generate the immune response because of the presence of antibodies in recipients of other blood types. All right. So, so we have lipids obviously undergo a lot of recycling, right? So there's constant dynamics, right? New cells are born, new membranes are formed, old membranes are destroyed. So lipids have undergo recycling, and there are a lot of enzymes which participate in the process. So specifically the enzymes, so the enzymes which break the ester bonds are known as esterases. Esterases. But those that break the esters of fatty acids, so those are known as lipases. Lipases. So, so phospholipase A1 breaks the ester at this position, carbon 1, phospholipase A2 and carbon 2. Now there's phospholipase C. This one already breaks the phosphor ester bond. And phospholipase D also breaks a phosphor ester bond here. Right, there are two positions here. On the side of the inositol and on the side of the glycerol. And this lipid is phosphatidylinositol 4,5-bisphosphate. So gangliosides are degraded via a series of enzymatic cleavages, and so it's important that the failure to correctly degrade gangliosides results in buildup of lipids in lysosomes. So so dysfunction known as lysosomal storage disorder. Now there's a whole bunch of those. There's a whole bunch of those, and you can see here. Let's say that this is ganglioside GM1. GM1, so this is sialic acid right there. Sialic acid. So this ganglioside, for example, in the first step, this galactose will be cleaved, right, by an enzyme known as beta-galactosidase. Then hexominidase A will cleave off the square here, which is the N-acetyl-galactosamine. Now, if this enzyme is absent or is mutated, that cannot perform its function, then this disease develops, right? If this one is absent, then this disease develops, and so on and so forth, right? So this one is known. So for example, this molecule with the glucose attached to ceramide, right? So here glucose will be cleaved off by glucose cerebrosidase, and this disease develops if this enzyme is absent or defective, and a whole bunch of others. And one disease, actually, we're going to talk. I actually uploaded a paper for us to look at. If we don't have a chance to look at it today, we will look at the paper on Monday. There's this disease develops, which is the inability to process very simple ceramide and sphingosine type lipids. All right. So other lipids besides the lipids that make up the membranes. Do you have any questions about the material so far? No? All right. So lipids which are derived from the steroid based on steroid nucleus. So these are hydrophobic, contain hydrophobic backbone, right? So there is a six-membered, and five-membered rings attached in this fashion, or fused in this fashion, right? And so this forms the steroid backbone, and sort of four fused rings. There is an alkyl side chain on the end, usually. And there's a polar head group. Now, polar head group, in this case, it's OH. It's not charged. So it's polar, but it's not charged. So it's still considered a lipid, right? Because it does have a lipophilic and hydrophilic portion, right? But the polar head group is not sufficiently polar to actually, for example, to make the membranes, for example, right? So lipid membranes, but it can be incorporated into lipid membranes. And if you remember correctly, if you remember, what happens to cell membranes if you get cholesterol incorporated into them? How does cholesterol change the properties of cell membranes?
It affects their viscosity.
Viscosity, that's right. And yeah, so it makes them more rigid, right? Makes them more rigid. And it's one way, for example, cell membranes can regulate their fluidity. All right, so cholesterol present in eukaryotic cells, but sterols actually are absent in most bacteria. So we obtain cholesterol from food. A lot of it comes from food, but also we synthesize it ourselves in the liver. So if you have cholesterolemia, right? So if you have high cholesterol in your detected in a blood test, just limiting cholesterol by not consuming it from food sources is good, but it will not help you completely get rid of cholesterol, right? Because a lot of it is biosynthesized. And the interesting thing is that the less cholesterol you get from food, the more of it is biosynthesized. So the trend is very interesting here, and you really have to work on this, consult specific sources, or talk to a specialist on how to do this properly. So by just cutting this out from food will not work in most cases. So cholesterol bound to proteins, so you know that they're transported through blood vessels in a protein bound form. So here low density lipoproteins, so these tend to deposit in clogged arteries, so it's bad, right? So high density are usually good cholesterol. This is bad cholesterol. So you want the ratio to be changed in the to benefit toward the high density lipoprotein. And many hormones are derivatives of sterols. So in general, if cholesterol has only one oxygen, right? So we looked at the structure just a moment ago, has one oxygen. So cholesterol serves as the starting material for the production of a lot of hormones. They're called steroid hormones, and usually through oxidation. So they many of them have sterol nucleus, right? But lack the alkyl chain. And obviously, they actually are more oxidized, they're more polar. Remember, the more you oxidize something, the more hydroxy groups, the more carboxylic groups, the more polar your molecule will be if you add more oxygens. All right. So male and female sex hormones. So let's look at their structures. So testosterone made in testes. So this is male sex hormone, right? Estradiol is the female sex hormone. Cortisol is the hormone, and aldosterone. These are produced by the adrenal gland, and this one is responsible for glucose metabolism, and aldosterone is responsible for, let's say, blood pressure, and specifically responsible for retention of sodium in the kidney, right? So you know that one way a body regulates its blood pressure is by releasing salt or retaining salt, right? So why is that? So if you retain salt in the bloodstream, does your blood pressure go up or down?
It goes up.
It goes up, right? So if you retain sodium, so if the kidney doesn't dispose of sodium well, and then why will the blood pressure go up?
Because of diffusion or osmosis.
Osmosis, that's right. So if you have high salt in the bloodstream, then the water from the tissues will come into the bloodstream, right? And there'll be higher volume of blood in the bloodstream, basically, and that will result in a higher blood pressure. Now, prednisone, so you know this participates in inflammatory response, and prednisolone. So the difference is this carbonyl here, right? So there's a ketone in prednisone.
Aren't prednisone and prednisolone also used as anti-inflammatories? I know they're steroids, but like in medical use, aren't they used as anti-inflammatories?
Yes. So they're also used as drugs, but they have natural source, right? So they're actually not artificial drugs. That's right. So they're actual hormones. Mm-hmm. So there are other kinds of biologically active lipids. So many of them play roles as signaling molecules between nearby cells. And there are also lipid-soluble vitamins. So many lipids are actually vitamins, A, D, E, and K. So this one is actually my favorite type of lipid, known as arachidonic acid. Arachidonic acid is the carboxylic acid with 20 carbon atoms and 4 double bonds. All double bonds are cis. Okay. And so this actually undergoes very interesting chemistry. It's oxidative cyclization chemistry that gives rise to a variety of molecules with different functions. So arachidonic acid is actually derived from a lipid, right? So there's an enzyme, phospholipase C, which actually, sorry. There's an enzyme. Is it phospholipase A2? I think it's phospholipase A2, PLA2. PLA2. I'll write it down, but if it's incorrect, I will correct myself in the discussion section. Phospholipase A2. So arachidonic acid then diffuses into the cell, undergoes a variety of transformations. So some of it will form prostaglandins, So these molecules actually responsible for a variety of effects, such as, for example, inflammation, fever, pain. We should add pain here. And so, for example, many non-steroidal anti-inflammatory drugs will actually block this this reaction, right? So there is this reaction is actually catalyzed by enzymes known as cyclooxygenases, COX. There's COX-1 and COX-2. COX-1 or 2. And so there are non-steroidal anti-inflammatory drugs. So for example, aspirin, for example, ibuprofen, Tylenol, right? So all those will inhibit the enzyme cyclooxygenase and production of prostaglandin. And that's why many of them are used as anti-inflammatory, antipyretic, pain reducers. Yeah, that's how I could call these drugs. Drugs for pain, I guess. So another type of structures which are produced from arachidonic acid are thromboxane A2, for example. So this participates in the formation of blood clots, a very powerful factor. Basically, it, again, it stimulates the aggregation of platelets. So we talked about platelets. Many of these produce, so arachidonic acid also gives rise to leukotrienes. Leukotrienes are molecules responsible for the smooth muscle contraction in lungs. So for example, in asthma, very important, right? Asthma. So, so there are a lot of drugs which are used to treat asthma, which block the production of leukotrienes from arachidonic acid. You can see, so it's a lipid, right? It's a lipid. It lives there in the cell membrane, but then when it's necessary, it leaves the cell membrane, undergoes the oxidation by this enzyme, and produces a whole bunch of molecules with lots of different functions. Any questions about the prostaglandins?
What did you say that COX-1 and COX-2 do again? Are those for pain relief as well?
So they are all participate in the formation of prostaglandins. Yes, inflammation, fever, pain. So there are many different kinds of prostaglandins. There is PE, PG, PF prostaglandins. There are different letters associated with each prostaglandin, and they're all responsible for a particular type of response. And also depends on what kind of cells they're produced in, right? So, for example, if they're produced in a neuron, then that will cause pain. If they're produced in the white blood cells, right, that will lead to inflammation and things like that. So, so these enzymes are the ones which are responsible for the production of these prostaglandins.
Got it. Thank you so much.
All right. Vitamin D regulates calcium metabolism. So now we know that vitamin D is good for calcium, and we know the sunlight is good for calcium, and specifically the reaction or this biosynthetic sequence that leads to the production of vitamin D is shown here, right? So it starts with the 7-dehydrocholesterol. So you can see in cholesterol, you only have one double bond. In 7-dehydrocholesterol, there's a second double bond at this position between these two carbons. And so whenever you have a cyclohexadiene like this, it can, with light, it can undergo bond cleavage. This is known as an electrocyclic ring opening. You don't study this in OChem 2. Electrocyclic ring opening. So it's not studied in the OChem2, but it's studied in higher level organic chemistry classes. And there's a second step, the migration of the double bonds. So the organic chemistry on the slide is not represented in all its beauty, but there's a second reaction here, migration of the double bond from between carbons 9 to 8 to carbons 10 and 19. And so now you have two more steps. So so this is done in the skin, right? So UV light, as you know, UV light cannot really penetrate very deeply into the tissue, right? It only hits the skin, cannot cross the outer surface of the skin inside. And so so this molecule has to come to the surface before it can actually undergo this reaction. But once this is produced, this goes back into the bloodstream, it's delivered to the liver, and in the one step, it's actually, it undergoes now. So there are two hydroxyls. One is introduced in the liver, one is introduced in the kidney. And I just realized I can't remember which one is first, which one is second. I used to remember, now I forgot. But it doesn't matter for the purposes of our discussion. So one is liver, the other is kidney. Liver first, kidney second. And you get the finally vitamin D, which is now responsible for calcium metabolism. Specifically, one of the main mechanisms is the acquisition of calcium through intestinal absorption, right? So basically it allows for intestines to absorb calcium from the food more easily. Okay, so vitamin A, another biologically important lipid. So this one is involved in the perception of light, right? So this whole thing starts from beta carotene, which undergoes oxidation, basically undergoes cleavage at this position, undergoes cleavage to give a molecule known as all-trans retinal. Retinal, you can see there's a whole bunch of double bonds. There's a whole bunch of double bonds, and it's an aldehyde. That's why it's called retinal. And it localizes in the retina. That's why it's retinal. And so there are actually two pathways for this. In one pathway, it can undergo oxidation to an acid to give all-trans retinoic acid, which is a hormone, and it's actually can change gene expression. And the second pathway for this is important for the perception of light. So it undergoes reduction to retinol, and then isomerization of the double bond between carbons 11 and 12 to give 11 cis retinal, and there's also oxidation again. So this retinol business going on back and forth all the time. So now this is 11 cis retinal, and this binds to a protein known as opsin to form a protein known as rhodopsin. So rhodopsin is a protein composed of the protein portion and the molecule of retinal. And that's the molecule which will absorb light. So remember I told you whenever you have highly conjugated systems absorb light in a visible region between 400 and 700 nanometers, right? And when it absorbs light, it undergoes isomerization to more stable transform. So remember, cis is less stable than trans. And so with the energy, if you add some activation energy to this process, it can undergo isomerization to give all-trans retinal. And obviously, if this is occurring within a protein, then the protein will change its three-dimensional shape. It will change its conformation, right? the protein will change its conformation, and that will generate the neuronal signal, which will then be propagated through a bunch of neurons to the optic nerve and result in the perception of vision, perception of light.
I have a question.
Yeah.
If the trans is more stable, why does it require energy to go from cis to trans?
Yeah, so this is activation energy. So excess of energy will be dissipated. Well, the rest of the energy will be maybe used for the change in the conformation of the protein, right? But in general, even though cis is less stable than trans, it doesn't mean that it will spontaneously undergo this kind of isomerization, right? Remember? So if this is cis, this is trans. Cis, trans. Like so, right? So you need activation energy to overcome the energy of the transition state, right? So it's not a spontaneous process. Does it make sense?
Yes, sir. Thank you.
All right. All right, and to finish off, what else do we have? We have vitamin E, K, and other lipid quinones, antioxidants. So vitamin E is an antioxidant. So this is shown here. So the way this usually works is this hydroxy group. Whenever you have phenolic hydroxyl, so compounds like that can serve as antioxidants, specifically because they can react with radicals, right? So for example, if you have a hydroxy radical, which is a highly damaging molecule, often referred to as reactive oxygen species, ROS, you may have heard, right? ROS. So ROS will remove a hydrogen from the phenolic hydroxyl to give rise to phenolic based radical like this. Like so, yeah. And this is a much more stable molecule. So if this can create a lot of damage, this one will not, right? And this will slowly find a way to neutralize this radical, whereas hydroxy radical can cause immediate damage, for example, to DNA, right? So vitamin E, antioxidant. Now vitamin K1, blood clotting cofactor, also known as phyloquinone. Also a lipid, warfarin, blood anticoagulant. So the question is, why is this a lipid? Not totally sure. We don't really have a lipid tail, right? So we have some lipophilic portions, but it might be interesting to look this up. Why is warfarin considered a lipid? Anybody knows? Ubiquinone. So this molecule is an electron carrier. Now it's based on a quinone, hydroquinone system. So before many of you were born, those of my peers and before those who were into photography, they were using film photography, right? And that's which was based on the redox process between the quinone and hydroquinone. So if this is quinone, it can receive 2 electrons. It can receive 2 protons. And go to hydroquinone. And the reverse process is also available, in which it will lose the electrons and the protons. Right? And so even though we use this in photography, but biological systems utilize this long before. We thought we invented this, right? And so in mitochondria, you know, mitochondria are the primary source. These are energy organelles, right? And so they will use the electron carriers, such as, for example, NADH, and they will, so these will be electron acceptors, right? Electron acceptors, and they can carry the electrons across the cell membrane. Plastoquinone, so this is very similar, only in plants, in chloroplasts. And this molecule is sugar carrier. So I guess this hydroxy group can be glycosylated. Okay, and there are other biologically active lipids. I think we're almost done. So polyketides. So these are diverse family of compounds synthesized similar to fatty acid biosynthesis. So these are the only reason why these are called maybe lipids is because because the process of biosynthesis is very similar to that of fatty acids. The specific characteristic feature of these polyketides is the location of oxygen functionality, whether it's a carbonyl or OH, at the sorry, not this one. Not this one. They are separate at the carbons which are separated by another carbon. In other words, position beta. Right, so these are beta carbons. And same thing here. So this is erythromycin. So you know that many of you have taken this as an antibiotic. Amphotericin B is also used for fungal infections. Lovastatin is a cholesterolemia used for to reduce blood cholesterol. And all right, you have a, so what did we learn here? Lipids, structurally, functionally diverse class of molecules, poorly soluble in water. Triacylglycerols, remember those on Monday, main storage lipids. Phospholipids, main constituents of cell membranes. And sphingolipids too, but they also play roles in cell recognition, specifically through their carbohydrate moieties. Cholesterol is both membrane lipid and precursor for steroid hormones. And some lipids, so we talked about those, right? So specifically like prostaglandins, for example, right? They carry signals from cell to cell and from tissue to tissue. All right, guys, do you have any questions about anything we've talked about today?
I have a question about the blood types. So isn't there an additional like subset of blood types is that just not based in the same format, and so that's why we didn't talk about it?
Yeah, rhesus, and yeah, so it's the presence or absence of this additional protein, Rh plus or minus. Is that important?
Yeah.
Yeah. So, but that has nothing to do with the lipids. It's just a protein. But
Okay.
so it's involved in further. The Rh factor also is important, and in pregnancies it's important. But that's just just presence of absence of a particular protein in the cell membranes.
Oh, okay. Okay, cool. Thank you.
41% of Americans vitamin D deficient. Get out in the sun. Yeah, just wear a mask. Sounds good. All right, guys. So on Monday, so this homework is due, don't forget, this homework is due on Saturday, right? It's not a long homework. So, and the material is pretty straightforward, mostly descriptive, no math, nothing really to think hard about. So homework due on Saturday, and on Monday we'll look at the paper that I uploaded and we'll start chapter 11. Sounds good? All right. I'll talk to you all on Monday.
Thank you.
Bye.